floor is yours.
Thank you. Good morning, everyone. So just before to start my presentation, I just want to say one very important thing for me, and of course for you, because I am deaf, and you can see, but I speak quite well. But sometimes it's very difficult for me to speak and to pronounce. So sorry if sometimes you don't understand me. So thank you. So to start. Okay. So to start today, we will speak about chromatin structures. And if you want to study chromatin structures with the wet lab method, it's very difficult because we can study at the same time a lot of genes in the space, 3D space. But with omic sequencing, it's become possible to study the 3D chromatin structures. For example, here, with these techniques, we can sequence the interaction between chromosomes. And for one wind, we have the two fragments from these chromosomes 1 and chromosome 2. And so it's possible to study the chromatin structures with the 3D chromatin network. And so we can reconstruct the 3D, the chromatin structure. Here you have an example that we can do with a GardenNet. Is a software we can reconstruct the chromatin structure. So here you have the chromosome. We map the reads on the human genomes, and we can reconstruct the 3D chromatin structure. So we need to find the interaction between the chromosomes, and we defined the chromatin assortativity. The chromatin assortativity identified what extent property of chromatin fragment is shared by fragments that interact frequently with it. And so if the chromatin assortativity is above than 0, so it's assortative. If the charge is equal to 0, there's no particular association between the node property. If the charge is below than 0, the network is dissociative. So now I will talk about cancer, because I would like to study the 3D chromatin structure in cancer. Cancer is a complex object. You have several cells. You have cancer cells. You have immune cells, which can recognize the cancer cell and kill them. And you have fibroblasts, or microenvironment cells. And for each cell types, we have a wide or narrow heterogeneity, which can explain drug resistance. And you want to avoid that. So understanding the cell phenotype is an important key if you want to overcome this disease. Which mechanisms are involved in normal and cancer cells phenotype and its heterogeneity? So you have several theories of cancers. Some are not very serious, like God can explain you have cancer. So no, it's not scientific. But you have someone is very interesting. It's called the atavistic. Sorry, I have some problem with the. So the atavistic theory of cancer. When life appears on Earth, you have unicellular cells. And these inner cells live in very dangerous environment. They need to be very plastic to adapt and to survive. But the first unicellular cells were prokaryotes without nucleus. And after you have unicellular eukaryotes with a nucleus, and the genome starts to be very well organized. After that, the cells start to work together, and so to form a multicellular organism. And so at this time, the cells became very well organized. Very well, we have distinct phenotype, and that occurs during the development. Okay, so here you have vascular gastrulations. At these times, around 1 billion of years, you have organogenesis and body plan. body plan, so you can have hand, foot, and so on. So you have different cells at the end, like neuron, columnar epithelial cells, red blood, and so on. But what is a cancer? You have a very well differentiated cells. Okay, but these cells try to become an undifferentiated cells, like at the origin of the life. Okay, that's the theory, the atavistic theory of cancer. How we can explain the cancer cell phenotype? Do you know the epigenome landscape? Here you have a figure. Here you have an undifferentiated cells. Here you have the times, developmental times. And here you have some valleys, mountains, and so on. And so here you have a cell type like neuron. Here is another cell type like hair cells. And here you have maybe the cell from the skins. Okay, but how we can explain these different cell types? Here you have genes, but these types have different chromatin structures. And this chromatin structure can be explained by the proteins like polycombs or histone mark. And this DNA methylation and RNA modification can explain the modification of the epigenetic landscape. Okay? So, the first study tried to prove the atavistic theory of cancer. They classified the human genes in 16 classes, from the old unicellular ancestor, early metazoan, and more specific, in 3 classes. And so they did, okay, we will compare the gene expression of these 16 classes with in 7 solid tumors and their normal tissue. And what they found, they use the transcriptomic age index to measure the expression level. And so a low TI corresponds to higher expression of genes from early genes. And here you have the 7th tissue. And what we observe that with tumors, the TI tumors is low. So you have higher expression of genes from early genes. This result shows that the atavistic cancer is not just a theory. Okay, we have 2 aims. The first is to study the inter-individual gene expression variability in immune cells. The question is, do we have a relationship between gene age and gene expression variability in immune cells? If yes, which epigenetic mechanisms are involved in gene expression variability? Why is it important? If we understand the mechanism of variability in immune cells, we can improve immunotherapy. And so, the second is study the 3D genome structures during differentiation in cancer. I will explain later. So we took the gene age classifications and we took a public data set from Blueprint with 125 healthy individuals and the monocyte. And we observed that the gene expressions decrease with the evolution in gene age, and the expression variability increase with the gene age. So we look at the epigenetic markers, and for the active epigenetic markers, they are all enriched in all genes. But if you watch repressive epigenetic marks, they are enriched in middle genes, in early metazoans. And this is, okay, in the proteins, they are enriched in, like polycombs, they are enriched in early metazoan. Okay, but now we check the methylation status. With the methylation score increase with the gene age. So newest genes in blue, they have highly enriched in CpG. But the methylation variability increases in newest genes. So you have a higher methylation variability in the newest genes. In conclusion, in unicellular ancestor, all genes they are enriched in active epigenetic marks. In early METAS1 middle genes, they are enriched in polycomb target genes. Polycombs, they modify the chromatin structures. And so in more specific, new genes that are enriched in DNA methylations. And we did a chromatin network. And what we observe that all genes are in the core of the network, and the newest genes are at the edge of the network. And we did that on the promoter-promoter network. So now we start to discuss about 3D genome structures during differentiation and cancer. Do we have a relationship between gene age and 3D genome structure? Why is it important? Because the epigenetic modification can explain the cancer cell phenotype and plasticity, and we hope to develop a future strategy to cut off the cancer development. So we have downloaded three public datasets. Is the human embryonic stem cell, which is undifferentiated. You have B cells, which is AC differentiated, and leukemia, which is cancerous. So we study the polycombs in the whole genomes, and they are in human embryonic stem cells and B cells and CLL. CLL is leukemia. So we have an increasing of polycomb in B cells. So we have during the differentiations, you have an increase of polycomb. And during the cancerogenesis, they decrease. But in which age classes? So they are only enriched in early METAS1. So it's logical because during this evolutionary age, you have the body plans and the organogenesis. And so you can define the cell type. So we started to study the 3D chromatin structures in a network. So here we have a network. We don't see, it's very small, but it's a big, huge network, and like that. So we have different core with all genes and middle genes, and so on. Now, I will compare the human embryonic stem cells. They are undifferentiated in the epigenomic landscape. If we can compute the CHAS, the chromatin assortativity, and they are highly enriched in all genes. But when we compare with the B cell, differentiated cells, they become differentiated, and we compute the CHAS. And so what we observe, the early METAS1 is assortative, like all genes. But now we compute we did the network for the leukemia. So it was very well differentiated cells, and they tried to move on to the hills and try to become an undifferentiated cells. And what we observe for the CHAS, but we observe that the early METAS1 genes decrease. So they lost the multicellular context. They changed the phenotype. So we did a chromatin network, but we can't validate our result with wet lab method. It's impossible. So we find another strategy. We work with Gustav Steingunper. He was a PhD student at the Pre-Westfield Labs. He's here, Darius. And he did 3D polymer representations of the nucleus. And so he used a spring model to exploit molecular mechanisms. So the genes are beads, which is represented in circles, and you have here a bound between the fragments. And without these interactions, we can change the interaction between these fragments to reconstruct in 3D the chromatin structures. And so we obtained the 3 chromatin model. So it's still a work in progress because we have little problems and we want to compare this 3D polymer representation with our chromatin structures. So in conclusion, variability of gene expressions is associated to 3D chromatin structure in monocyte. So in terms of chromatin 3D structure, we think evolutionary older genes are found in the core of the chromatin network distributed in set of core, also broadly corresponding to the less variable genes. We identify consistent change during oncogenesis in the partial organization of these genes, maybe specific evolutionary class, reinforcing the important role that all genes and their organization in the nucleus play in cancer phenotype. This finding suggests the potential antibiostic theory confirmation at the level of chromatin organizations. Thank you for your intentions. And for the question, you can ask me question, but some people will help me to understand you because it's really difficult for me.
Thank you very much. Are there any questions for Benoît?
So I was wondering if you had different ways of building the networks, different parameters, and if it impacts the results. Je peux le faire en français. Est-ce que vous avez différentes façons de construire les réseaux ou différents paramètres et est-ce que ça impacte les résultats?
Yes, we have different methods to construct the chromatin network. So we can use a 3D chromosome. You can use a 3D dataset from the P chip or IC, and after that we can add another dataset like ChipTech to construct our models, and we can use different random to compute the class and to construct the model.